Thanks a bunch and have a great conference.
Credit card ready. Awesome. So our next element on Exponential sort of 101 is the field of regenerative medicine, stem cell biology and beyond. And really pleased to bring Nina Tandon, who is the founder and CEO of EpiBone, who's going to talk about regenerative medicine and the world of biological fabrication. Nina. Thanks.
Thank you. Hi, everyone. Having a good day? Good. So I'm going to invite everyone to start off by closing your eyes for a moment and scan your body. Feel your bones supporting you in your chairs or your bean bags, as the case may be. We are each made out of 206 bones held together by 360 joints. But many of us are more than that. You can open your eyes. By the time we go through this lifetime, 75% of us are going to be living with parts of our bodies that we were not born with, if we aren't already. These are the dental implants, the IUDs, the pacemakers, joint replacements that we tend to accumulate over a lifetime. My world is in skeletal reconstruction. Bone is the most transplanted human tissue after blood. And we are replacing millions, over 1 million joints every year in this country alone, just because of a couple millimeters of damaged cartilage. Okay, welcome to the $100 billion medical device industry. And you know, the medical device industry has made incredible strides. Things like insulin pumps, organ donation. You might even put in there, you know, just medical science has made a lot of strides at kind of creating an almost one-stop body shop for human parts, whether those parts are borrowed from donors or engineered from scratch. We've made incredible strides, so I don't want to belittle that. And yet, sometimes it's hard, you know, I wonder, have we lost our way? You know, the field of transplantation science has saved lives, but has led to shortages of organ donors. You know, medical devices have, you know, allowed us to replace joints, but then those joints need replacement, especially for our younger patients. If you're under age 60 and you get a knee replacement, you're likely to need revision surgery in under 5 years. Okay, and so it almost reminds me of this, you know, when I think about, you know, just taking a step back and thinking about our trajectory as a species and how we view the body. Through so much of history, I call this Body 1.0, you know, and I love this image of Leonardo's Vitruvian Man. It sort of evokes this idea that for so much of human history, medicine really was about letting the body come to rest, because there was a sort of assumed implicit, how do you say, proportionality and sense attributed to the body. But somehow around the turn of the last century, when we started developing things like interchangeable parts on the assembly line, we started to view our bodies in a similar way. And this is why I think of C3PO. If you needed a new part, you might get one from a donor, or get a bit and bob made out of metal or plastic. But we were thinking of ourselves as if we were like machines, and therefore the medical industry has sort of been like mechanics in a way. And you know, I love the previous speaker, you know, because we're starting to realize now, you know, the Human Genome Project and advances in biology have led us to realize that actually maybe more like interchangeable parts, we're actually drilling a level deeper into our understanding of the body. And our understanding of the body as if we are an ecosystem now of cellular beings, trillions of cells, most of them not even human, conspiring to create these bodies that keep us alive and sustain what we call a lifetime. And I call this Body 3.0. I really like this, this development in our understanding of the body. You know, it's led to the CAR-T therapies when we think about fighting cancer with using our own cells. It's led to my field of regenerative medicine, where we say, let's take the stem cells that grew our bodies in the first place and repair our bodies every day and collaborate with them to make new body parts. It's led to the understanding of the microbiome and thinking about bugs as drugs. So this is a really interesting kind of evolution in our thought that reflects a deeper understanding of complex systems. And it's almost as if we finally realized that man is a modular system, humans. And cells are the pixels in this world. I'm co-founder and CEO of a company called EpiBone. We apply this principle towards transforming patient lives through personalized skeletal reconstruction. We use digital fabrication in order to get the right shape, and we use stem cells because we're creating living implants. Here's a short video explaining our process. We take a CT scan from which we can extract three-dimensional data. From that, we can create personalized scaffolding and bioreactors to cultivate the tissues. We take adipose tissue from patients from which we extract the mesenchymal stem cells, infuse them onto that scaffold inside our bioreactor. Our bioreactor is an advanced cell culture system that mimics the conditions of the human body, providing controlled oxygen, nutrients, and mechanical forces that those cells need to not just attach to the scaffold, but proliferate within the scaffold, differentiate into osteoblasts and chondrocytes, and lay down new matrix. And after 3 weeks, we have a piece of tissue that's ready for implantation. After implantation, this is what we've seen in our animal studies, large and small animals, that the host tissue and our graft integrate seamlessly together, vascularize, and integrate over time, becoming seamlessly indistinguishable from the body's own tissue. We've done this for bone, cartilage, combinations thereof. We're hoping to start human clinical trials next year. And it's a very exciting time for us. We're based in Brooklyn. If anyone's in town, give us a ring and stop by. There are many benefits to this type of approach, this living approach to repair. Firstly, because we use a perfect puzzle piece shape, it fits. Secondly, because it's your own tissue, there's no risk of rejection. And thirdly, because it's alive, these tissues can truly integrate with the body and hopefully need less revision surgery than these metal and ceramic implants do over time. But as we know, and as we heard also in the last talk, we're also going to need to make sure that these types of technologies resonate across the value chain. They have to result in savings on some level to the whole system as a whole, resulting in hopefully simpler, faster surgeries for surgeons, shorter and easier recovery time for patients, fewer complications and revisions, and ultimately cost savings. Personalized medicine should not be more expensive medicine to the system overall. And so I just wanted to give you a view of some things that are happening in regenerative medicine as medicine is being redesigned on the cellular level. One of the goals, of course, is to make organ donation lists go the way of the telegraph. I couldn't resist a shameless plug for my own Ph.D. research. This is about a 10-year-old video. My work in cardiac tissue engineering, using electrical signals to get those cells to beat. Cardiac tissue is a pretty far way off from being able to create organs that are ready for implantation. But they are doing quite well as surrogates for testing drugs in the lab. Tony Atala and his group at Wake Forest University, they implanted their first patient with a tissue-engineered bladder in, anyone know the year? 2011, okay? So this has been happening for a little bit. This is Laura Nicholson's company called Humacyte. They're engineering vasculature in the lab, and they're in phase 3 clinical trials right now and hoping to file for a BLA next year. So this is progressing. EpiBone, we're hoping to begin human clinical trials next year, add 5 years, and then hopefully we'll have a product. And United Therapeutics is on the more complex end of the scale. When you think about engineering organs and parts, right? What's a simple tissue? Something flat, single cell type, something like skin, right? Then you've got hollow organs like the bladder and blood vessels. And then on the other end of the spectrum, you've got the heart, the lung, the perfused organs that are very complex, very greedy tissues. And so what United Therapeutics is doing, huge admirers of their work, is starting to now invest in lung printing, tissue engineering. Cellular therapies also can involve using bugs as drugs. This is like the microbiome. And there's so many samples of really good projects out there. I just want to highlight the work of Sangeeta Bhatia from MIT, where they've actually engineered, using genetic engineering, bacteria to glow green in the presence of colon cancer cells. Okay? So you can imagine a probiotic yogurt being a kind of diagnostic device in the future, using these little micro machines called bacteria. This is from Emulate Bio, a company that many people in this audience probably know quite well, where they're miniaturizing organoids on tissue chips that can help disrupt clinical trials. So, a lot of this motivation that came from medicine has really allowed us to think about cells as living building blocks, has allowed us to understand so much about how cells work and what they do, and has almost led to a bit of a paradigm shift, which was alluded to a little bit in some of the previous talks, where one could almost ask a question, can we do fill in the blank with cells? And surprisingly, there are a lot of answers to that question that are yes, in fields that you wouldn't expect. Okay, and so I think that what's really interesting is that some of this work in medicine is leading to a now broader field called biofabrication, okay, using biology as a mechanism for making things. This is from a company called CallPlant. They're based in Israel, and I gave the example of United Therapeutics working on printing lung, right? What these guys are doing is they've actually engineered tobacco plants to produce recombinant human collagen, okay? So using a plant as a plant, like a power plant type plant, like a factory. And they have a collaboration, we should read the press release from United Therapeutics, where they have an exclusive license for anywhere in the universe, for not just global, for this collaboration. Okay, these are examples from two different companies. One was mentioned in the last talk on the left, Modern Meadow. They've developed a biomaterial called Zoa that it's kind of like a leather that's made from producing collagen using these types of techniques in the lab. This is actually an exhibit from the MoMA, these two examples. On the right is a picture of silk that was engineered using yeast. And that design is by Stella McCartney in a collaboration. Okay, so fashion is being influenced by these types of techniques. Textiles. This is a company I love called Executive. They use mushrooms to engineer materials that can be used in furniture or packing materials. This is Marjolein Helder. She is the CEO of a company called Planty. She's figured out in the Netherlands how to extract some of the electrons that are released by plants as part of photosynthesis into the soil and use those for generating electricity. Again, like a power plant made of plants. This is Ginger Craig Dossier. She's got a company called Biomason, where they've developed a kind of concrete in which microbes are embedded. And when that concrete is exposed, it has cracks made in it, and those cracks are exposed to water. Those wake up those bacteria to self-heal the concrete. Okay, so we have bioactive concrete that can be kind of like a living architectural element. This is from a company called Koyo here in California, on the other end of the design life cycle, incorporating mushrooms that the designer J-Rim Lee calls the infinity mushroom, that can help remediate the toxic byproducts that are released by the body after death. So she's kind of made this living coffin, so to speak, that can help aid the decomposition process. And a fun example, if that was a downer for you, this is an example of taking microbes and extracting image data from them under a microscope and using them as pixels in a living video game. This is from Ingmar Rydell Cruz's lab at Stanford University. It was really fun for me when I was part of the Community Bio Initiative at MIT to work with students in biohacking communities around the world, making these biotic video games. In some cases, students were going to the pond and getting microbes from the pond as opposed to going to Carolina Biological Supply. It was really fun. But yeah, I think a lot of people are starting to see the parallels between the semiconductor industry of 40 years ago and where we are with our understanding of biology and how to interface between biology and traditional modes of fabrication. So what's next? You know, we're now learning to program microbes, program human cells. This is an image from MIT Technology Review when the first programming language for bacteria was released. Bacteria, or cells in general, as we can learn to reprogram them, can become even more flexible collaboration partners as we think about fabrication in the future. And when we think about scalability of these types of technologies, it's really important for us to think about automation of cell culture. And robotic cell culture is going to be a huge part of that type of scalability. But in the last few moments that I have with you, it almost begs the question, what can't be done? Right? And some of you are scratching your heads. How many people have seen this image before? Quite a lot, right? This image is so iconic. It's from the late 90s, I believe. And it's not quite clear, no offense to the Vacanti lab, please, I hope. But it's not quite clear why this rodent needed this ear on its back, and so I think it was so evocative for so many people, and actually inspired 2 artists to enroll at Harvard University to, as research fellows, to try and learn the skills of tissue engineering so that they could make bioart. Okay. Bioart, I think, has a capacity to help us be, as a community, self-reflexive in terms of where we're going as a field. And I think, are we tinkering with life for the purposes of education, for the purposes of entertainment, or for the purposes of medical science, right? Is tinkering with the DNA or genetic material of plants, bacteria, yeast, humans, rodents equivalent? Right? And if you can take the cells out of my body, grow them in the lab, put them back in my body, and the cells don't know the difference, and if our bodies are really made out of 10 times more bacterial cells than human cells anyway, what exactly are the boundaries of the human body anyway? There's some really interesting questions that come up when we start thinking about these technologies, and I think it's really important for us to consider the bioethical considerations that arise. And when many people think of bioethical considerations, we think of designer babies or body modification. But I also think that it's important for us to note, or ask the question, especially in this community, are the modalities of government grant funding, angel funding, and VC really incentivizing us to develop the technologies that we really want to see? Are they? I think it's an important question for us to ask. This is the valley of death. This is where a lot of technologies die on the vine because of that gap in feasibility and funding between those different types of funding sources. And I think it's also really important to note that access to these types of next generation technologies is as much a bioethical consideration as designer babies can be, and body modification. And so here's some data just showing the Healthcare Access and Quality Index from 2016, and you can see that we still have a long way to go. The first industrial revolution, before the first industrial revolution, there was a very little disparity in the human lifespan across the world globally. The industrial revolution created a huge spread there, and we run the risk, if we don't do this right, of creating an even wider disparity. So I love this quote from Anton Chekhov: "The role of the artist is to ask questions, not answer them." And so I think, in part because I believe the body is a work of art, but also that artists play an important role in our society, I wanted to end by showing you an image from our artist residency, Eliza Stamps. This is an image that she calls 170 hours, which is a time point that corresponds to how long it takes for human mesenchymal stem cells to show the signs of bone differentiation. And what I found out later is that that actually does correspond in certain studies of education to how long it takes for a human to really incorporate and learn a new idea. So, in closing, I'd like to invite you to, again, scan your bodies and imagine a future in which all those spare parts that we're bound to have accumulated in the 115 years that we all plan to live are made not out of metal, not out of ceramic, not out of parts carved out of yours or other people's bodies, but made out of our cells. I hope that I've given you a flavor of how these types of techniques that are used in medicine are disrupting whole industries as far as architecture, art, and energy. And I'd like to end with a provocative question, if you allow me. If the first industrial revolution was about machines, And the second industrial revolution, which you could argue we're in the middle of right now, is all about data. Wouldn't it be nice to think that the third industrial revolution could be about life? So, thank you very much. Oh.
Thank you, Nina. Awesome. I always get asked the question about when we'll be seeing complex 3D printed organs. Do you think that's going to happen, or is it like it gets supplanted by xenotransplantation from humanized pigs?
I think they're complementary technologies, but the thing about the xeno approach is that there's so much of the organism that we don't quite know how to recapitulate yet. And so having a living bioreactor, so to speak, these pigs, that, in my view, could be a faster approach. I think they're parallel processes that should be explored, and I'm definitely watching that field very closely and rooting for them.
Right. George Church, who spoke here 3 or 4 years ago, said clinical trials in that realm is going to happen in the next 2 to 3 years. Mm-hmm. And, it's already in primates.
Yeah, but tissue engineered approaches are probably 10 years away.
All right. Thanks, Nina.
Thank you.
All right. So if your brains aren't full yet, we need to accelerate our ability to learn